Vibrio fischeri as a host for genetic engineering, Oil Degradation Bacteria, Alcanivorax borkumensis, by Michael Fraczek, Players K-12, Anchorage, Alaska. Every oil spill in a marine environment threatens the very nature of that ecosystem. This project investigates a method to remediate ecosystem damage as quickly as possible. According to the National Oceanic and Atmospheric Administration, over the past 50 years, our environment has been damaged by at least 44 different marine oil spills, with each oil spill dumping an average of 4,200 gallons of crude oil into the Earth's oceans. An example includes, on March 24, 1989, the infamous Exxon Valdez spilled 42 million gallons of crude oil into the Prince William Sound in Alaska. The oil spread 1,990 kilometers of the coastline, killing over 250,000 marine organisms, leaving long-term effects from the oil contamination still present today. Another example, on 2010, Deepwater Horizon spilled an estimated 171 million gallons of oil into the Gulf of Mexico, covering a total of 1,100 miles of coastline, 200 square miles of ocean floor, and 68,000 square miles of surface water. In the process, this spill killed millions of marine life. One way to mitigate oil spills is to use bioremediation. Bioremediation uses naturally occurring microorganisms to consume environmental toxins. According to the U.S. Geological Survey, there are many instances when these microorganisms have been successful in various gasoline and crude oil contaminations over the past several decades. Various bacteria species play significant roles in degrading crude oil, an important example being Alcanivorax. More specifically, the bacteria in the genus Alcanivorax demonstrated the largest growth in oil spills, showing that this bacteria can thrive off crude oil. The original idea for this experiment was to take a bacterium, Alcanivorax borkumensis (SK2), and amplify its ability to be used as a method of bioremediation for crude oil, specifically various mixtures of N-alkanes. Targeting N-alkanes is important because they make up about 55% of crude oil, along with aromatics at 20% and polar molecules at 13%. However, research showed that this bacteria was both hard to cultivate artificially in the lab and difficult to genetically manipulate. Additionally, this bacteria is also expensive, especially for continuous repurchase, and not extensively documented, creating the need for a separate host for this experiment. In response, a separate marine bacterium, Vibrio fischeri, was chosen for genetic transformation of the genes responsible for A. borkumensis. This bacteria was chosen for its inexpensive price, as well as its well-studied habitat range. V. fischeri lives in both temperate and tropic waters, as both a free-floating bacteria and a symbiotic partner to the tropical Hawaiian squid shown on the right. It is also relatively easy to culture. A marine Arctic bacteria, Polybacter argensi, was another idea for this host bacterium, but due to its limited availability in different oceans, it was not as favorable as an option. Research indicates that there are 40 or more genes involved in alkane degradation. Additionally, multiple separate clusters of genes are located apart from each other on Alcanivorax borkumensis genome that all cumulatively allow this bacteria to terminally oxidize a wide spectrum of alkanes. So using the research, the targeted genes and operon is shown above in the red box. For this year, the focus is researching A. borkumensis and V. fischeri, as well as running preliminary tests that stand as precursors to the actual experimentation of this project. For next year, and possibly the year after that, using polymerase chain reaction on A. borkumensis to splice and amplify a certain gene of choice. Then I will use restriction enzymes and DNA ligase to insert the gene into a certain plasmid containing a specific antibiotic resistance gene, which will hopefully be transformed into V. fischeri and grown on a selective medium. Finally, I will test to see if the experiment was successful. Therefore, the purpose of this project is to genetically engineer Vibrio fischeri with alkane oxidizing genes from Alcanivorax borkumensis, with the goal of making Vibrio fischeri terminally oxidize N-alkane C5 to C12 found in crude oil. My hypothesis is that if the genes from Alcanivorax borkumensis are genetically engineered into marine bacteria, Vibrio fischeri, then N-alkanes C5 to C12 will be oxidized by Vibrio fischeri. There have been 3 preliminary tests completed over the past few months. The first preliminary test compared V. fischeri's growth on Photobacterium agar and marine agar, both known growth mediums for V. fischeri. The second preliminary test compared the growth of V. fischeri on nutrient agar, more economic agar, and marine agar with ampicillin to test its resistance to this antibiotic, all against a known positive control of V. fischeri on marine agar. As for the third preliminary test, the ability to keep Vibrio fischeri in a glycerol stock for long-term storage is explored. Results for the first preliminary test, the qualitative data of the 4 petri dishes show that there is more noticeable bacterial growth on marine agar over Photobacterium agar. For the third preliminary test, although there was growth on the nutrient agar plate, due to the uncertainty of the white spots found on the plate, the test must be repeated for confirmation. For the marine agar and ampicillin plate, there were some colonies present to say that V. fischeri is resistant to ampicillin. Additionally, a study by the National Center for Biotechnology Information concurs about V. fischeri's resistance to ampicillin, as well as other penicillins. For the third preliminary test, the results are not ready by the time of this presentation's completion. However, the results will be ready by the time of Bio Expo for presentation. The next steps of this project is described above. The plasmid is also shown above with the tetracycline resistance gene. The gene of choice for this experiment is the operon previously described. If successful, this modified V. fischeri will degrade C5 to C12 alkanes in a lab setting. Future experiments include working with genes from recently isolated bacteria that's unattainable that allows for the degradation of long-chain alkanes from C15 to C30. These are the picture sources used in this presentation.